What if the foods you eat every day aren't just affecting your weight or energy, but quietly helping damaged cells survive and multiply? This isn't about fear, it's about biology. Certain common foods can create the exact internal environment that cancer cells need to grow. Today, we're breaking down the science behind the title, These Foods Quietly Feed Cancer Cells. Not myths, not panic, just mechanisms, and what you can do starting this week. And once you understand what cancer cells actually need to survive, many everyday food choices will start to look very different. My goal is to show you the mechanisms, the level of evidence, and practical steps you can take right away to shift the balance away from an internal environment that favors tumor growth. If you want more practical, evidence-based tips like this, subscribe now so you don't miss our follow-up deep dive on artificial sweeteners and cancer risk. First, let's look at a less obvious threat hiding in pantry staples, aflatoxins from contaminated nuts and grains. Aflatoxins are toxic compounds produced by certain molds that can grow on crops like peanuts, corn, and tree nuts when they are stored in warm, humid conditions. Mechanistically, aflatoxins are genotoxic. Lab models clearly show they can damage DNA and promote liver cancer, and epidemiological data in regions with high contamination levels link aflatoxin exposure to higher liver cancer rates. There's also a downstream pathway worth noting. Animals that eat contaminated feed can secrete aflatoxin metabolites into milk, so dairy products can carry a trace of the same toxin in the form of aflatoxin M1. Here's the catch. The risk is not uniform across the globe. In places with strict food testing, the levels in commercial products are usually low, while in regions with poor storage and regulation, the burden is much higher. Practical mitigation is straightforward and low cost in most settings. Buy nuts and grains from reputable suppliers when possible. Check packaging dates and storage recommendations. Roast or cook nuts before eating. Heat reduces some molds and toxins. Store them sealed in cool, dry places or the refrigerator if you live in humid climates. And prioritize tested or regulated dairy.
Quick tip. If nuts smell off, show discoloration, or taste bitter, discard them. Those are signs of spoilage that shouldn't be ignored. Also remember that nuts offer cardiometabolic benefits in many studies, so the goal is not to demonize them, but to reduce contamination risk while keeping their benefits. Next, let's talk about sugar and high glycemic processed carbohydrates, a category that directly satisfies the fuel need for rapidly dividing cells. Cancer cells often favor glucose and burn it quickly, a phenomenon called the Warburg effect. In simple terms, many tumor cells rely heavily on glucose even when oxygen is available. Mechanistic experiments in cells and animals clearly demonstrate that glucose fuels proliferation, and higher circulating insulin and IGF-1 levels act as growth signals that can amplify that effect. Human evidence comes largely from observational studies showing higher risks of several cancers, including breast, colon, and pancreatic cancers, associated with diets high in refined carbohydrates, sugary beverages, and insulin resistance. It's important to be precise. Mechanistic data are strong in lab models. Human epidemiology shows associations that are consistent with those mechanisms, but confounding factors exist. What you can do now is practical and achievable. Replace sugar-sweetened drinks with water, unsweetened tea, or sparkling water. Favor whole foods that release glucose slowly, legumes, whole grains, non-starchy vegetables, and berries, which provide fiber that blunts blood sugar spikes and supports a healthier gut environment. Portion control and regular meals that combine protein and fat with carbohydrates also reduce large glucose surges. Why that matters? Lower spikes in blood sugar and insulin mean fewer of the hormonal growth signals that can tip the balance in favor of mutated cells. Now consider processed and red meats, a food group that often comes up in discussions about cancer risk. The concern here connects to several mechanisms. First, many processed meats contain nitrite and nitrate preservatives that can form nitrosamines, compounds that are known to damage DNA in lab models. Second, high-temperature cooking methods like charring or pan-searing create heterocyclic amines, or HCAs, and polycyclic aromatic hydrocarbons, or PAAs, which are carcinogenic in animal studies and mutagenic in cells. Observational human studies, particularly for colorectal cancer, consistently show higher risk with greater consumption of processed meat. For red meat, the association is more nuanced and varies by total dietary pattern and other lifestyle factors. We must be cautious. Observational studies can be influenced by confounding behaviors. People who frequently eat processed meat may also smoke, exercise less, or consume fewer fruits and vegetables. Still, the mechanistic plausibility combined with repeated human associations led expert panels to recommend limiting processed meats. Practical steps are again realistic.
Cut back on processed meats like bacon, hot dogs, and many deli meats. When you do eat red meat, choose leaner cuts and limit portion size. Avoid charring and very high-temperature cooking. Use oven baking, braising, or lower-temperature grilling, and trim charred bits before eating. Pairing meat with fiber-rich vegetables and whole grains helps dilute potential exposure and supports a protective gut environment. Another layer that often gets overlooked is chronic low-grade inflammation. Dietary patterns high in refined carbohydrates, trans fats, and ultra-processed foods can keep inflammatory signaling slightly elevated for years. That matters because inflammation releases cytokines and growth factors that help mutated cells survive, invade surrounding tissue, and evade immune detection. Large cohort studies consistently link inflammatory dietary patterns to higher risks of colorectal, liver, and breast cancers. In contrast, diets rich in fiber, omega-3 fats, and polyphenols are associated with lower inflammatory markers, shifting the internal environment in a less permissive direction. Alcohol deserves its own careful attention because the evidence level for harm is comparatively stronger. Alcohol is classified by the International Agency for Research on Cancer as a Group 1 carcinogen. Mechanistically, ethanol is metabolized to acetaldehyde, a compound that can damage DNA and interfere with DNA repair processes. Alcohol also promotes inflammation, can worsen nutrient deficiencies that are important for DNA repair, folate for example, and acts as a solvent that increases absorption of other carcinogens. The strongest causal links are for cancers of the oral cavity, pharynx, esophagus, liver, and there is convincing evidence of increased breast cancer risk even at moderate intakes. The public health message here needs nuance. Lower consumption reduces risk, and for people at particularly high risk, strong family history, or existing precancers, abstaining may be the safest choice. Quick mitigation is clear. Limit intake to the lowest level possible, and understand that a drink a day is not risk-free. Combining alcohol with tobacco multiplies risk, so avoiding both together is especially important. There's one more biological layer that ties many of these foods together: excess body fat. Adipose tissue is hormonally active. Higher levels of body fat increase estrogen production, insulin resistance, and inflammatory signaling, all of which can promote the growth of certain hormone-sensitive cancers. This helps explain why long-term metabolic health matters more than any single food. Throughout these blocks, remember that the effects add up. A diet that consistently produces high glucose and insulin, contains frequent exposure to processed meat-derived compounds or alcohol, and is low in fiber and antioxidants, creates an internal environment more permissive to tumor growth.
Conversely, dietary patterns rich in fiber, varied plant foods, and minimal processed items tend to reduce inflammation, stabilize blood sugar, and support immune surveillance. That isn't a guarantee of prevention. Genetics and chance matter, but shifting patterns reduces probabilities over time. So what practical, prioritized actions should you take this week? Check how you store nuts and grains. Keep them sealed, cool, dry, and use older packages first. If you buy in bulk, split into smaller airtight containers and refrigerate in humid climates. Roast or cook nuts when appropriate, and prefer suppliers that use food safety testing if you can. Cut sugary beverages and reduce servings of high glycemic foods. Swap a soda or sweetened coffee for water, unsweetened tea, or a small serving of whole fruit. Aim for meals that combine fiber, protein, and healthy fats to blunt glucose spikes. Reduce processed meat intake. Treat bacon and sausages as occasional items rather than staples. And when you eat red meat, favor cooking methods that limit charring and balance the meal with generous vegetables and whole grains. Reassess alcohol habits, lower frequency and amount, and avoid drinking to intoxication. If you have personal or family risk factors, talk with your clinician about whether abstaining is advisable. Prioritize fiber. Beans, lentils, vegetables, fruits, and intact whole grains not only lower glycemic response, but also feed beneficial gut bacteria that can support immune function and reduce inflammation. These 5 steps are not dramatic overhauls. They are modest, cumulative changes that shift your internal environment away from what cancer cells find favorable. A few nuanced points worth underscoring. Nuts, for example, are complex. Many studies show cardiovascular and metabolic benefits from regular nut consumption, so it would be misleading to label them categorically dangerous. The concern is contamination in specific contexts. The response is targeted, improve storage and sourcing, rather than elimination. Similarly, processed meat carries more consistent signals of harm than unprocessed red meat, but overall dietary patterns matter most. And when I say sugar feeds cancer cells, I am not implying that every sugar calorie you eat will cause cancer. Rather, persistent high blood sugar and elevated insulin create an environment where pre-malignant cells are more likely to survive and expand. For the evidence-minded viewer wondering about the strength of claims, much of what we discussed rests on a combination of mechanistic lab data, animal studies, and observational human research. Mechanisms such as the Warburg effect, acetaldehyde's genotoxicity, and formation of HCA's PHAs at high heat are well described in experimental settings. Human data, especially for alcohol and processed meats with colorectal cancer, are consistent and strong enough for public health recommendations.
For other links, like sugar and specific cancers, human evidence is associative with supportive biological rationale. Randomized trials on cancer outcomes are challenging and rare, so we rely on the totality of evidence and plausible mechanisms to guide practical advice. You may wonder about practical limits. Do you need to shop expensive brands or overhaul your kitchen? Not at all. Many of the protective steps are low cost: choosing water over soda, increasing beans and vegetables, storing foods properly, and choosing cooking methods that avoid direct charring. If you're concerned about higher risk exposures because of where you live or your occupation, discuss targeted testing and personalized strategies with your healthcare provider. Before we finish, a brief note about what we're not covering today. Artificial sweeteners and their cancer risk remain controversial and deserve a focused examination. I'll have a follow-up video unpacking the latest evidence so you can decide how they fit into a lower risk diet. If that interests you, make sure you're subscribed so you don't miss it. In summary, cancer cells need fuel, growth signals, immune permissiveness, and blood supply, and certain everyday foods and behaviors can subtly contribute to those needs. That doesn't mean a single bite will make or break you, but patterns matter. Prioritize fiber-rich, low-glycemic carbohydrates, reduce sugary drinks, limit processed meats and high-heat charred cooking, minimize alcohol, and practice sensible food storage to lower aflatoxin risk. Small, consistent changes reduce risk over time. If this was helpful, like and subscribe, and check out the playlist linked to learn more about practical nutrition moves and the upcoming deep dive on sweeteners. For concerns about personal cancer risk or before making major changes to your diet in the context of treatment, talk with your clinician. They can help translate these general strategies into a plan tailored for you.